Hello, and thank you for joining us today to learn more about the latest version of DILIsym software. Parts of today's session has been pre-recorded to optimize time. The video's audio feed will only be played through computer audio, so your mic and speakers. If you have dialed into the audio conference using the phone, you will hear the video play through your computer or your device's speakers rather than over the telephone. If you are not hearing the audio properly, whether you're connected to the computer or phone audio, please check the volume on your computer speaker or the device output. It is possible that your audio is playing through a different output, like a USB headset that's plugged in. But if you need assistance, please use the hand-raising icon. We take your privacy rights seriously, and by attending this event or participating in the Q&A session, you're allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. The Q&A will immediately follow the software demonstration. Now, before we begin, please tell us a little bit about yourself. Have you used DILIsym software before? We'll give you just a couple of minutes here to answer. Again, thank you for joining us, and those who have just tuned in, we're taking a quick poll to tell us a little bit about yourself. I'm going to close the poll now, and it looks like a large majority have never seen or used the program. So we're glad you're here with us today. Now let's get on to the powerful science. It is my pleasure to introduce the president of DILIsym Services, Brett Howell. Take it away, Brett.
Thank you, Arlene, and thank you everyone for joining us today. We're excited to unveil DILIsym version 10, or DSX. Before we get started with DSX, I wanted to mention briefly our company, Simulations Plus. DILIsym Services is a division of Simulations Plus, and at Simulations Plus, we have a powerful grouping of science, including chem informatics or machine learning, PBPK or physiologically based modeling, pharmacometric or POP-PK modeling, as well as quantitative systems pharmacology and toxicology. And it is this set of different types of modeling and simulation techniques brought together at Simulations Plus that makes us a unique provider of products and services. You can see here on the next slide that we have a number of available products. Some of these icons are very probably recognizable, including our GastroPlus platform, our ADMET Predictor platform, our recent acquisition of the Monolix suite from our Lixoft division based in Paris. So we have a great lineup of products, including the DILIsym software platform that we'll be discussing today. Today, you'll hear from myself, who Arlene has already mentioned, Brett Howell, as well as the star of the show, Corey Berry, our senior software engineer. So I'm going to mention a few features of DILIsym, including a brief introduction as well, and then I'm going to let Corey take it away for most of the webinar. First of all, for those of you who are less familiar with DILIsym and quantitative systems toxicology in general, I wanted to mention what makes up a quantitative systems toxicology model. We start with a mathematical representation of the relevant biochemistry. For today's webinar, of course, the liver and the biochemistry of the liver is most relevant and most important. We layer on prediction of the exposure of the compound, in our case, typically through our flagship GastroPlus product. And lastly, we introduce the specific toxicity mechanisms that intersect with the biochemical pathways and can lead to problems. This combination of things leads to the prediction of toxicity. Now, a quick overview of our DILIsym platform. Of course, it has a brand new interface and graphical user environment, and Corey will be discussing that today. In terms of science, we rely primarily on three key mechanisms in the software. That is the generation of or production of oxidative stress or reactive oxygen species, the effect on mitochondrial function and the resulting dysfunction, and the disruption of the transport of bile acids and phospholipids via transporter inhibition. So those mechanisms come together to cause injury, which can lead to inflammation and can also lead to regeneration and recovery of the liver in the software as well. We've now run the software on over a large number of validation or optimization compounds, and we've seen a high level of success. A lot of learning has come through those projects, and we've also begun using using the software for a lot of combination projects as well. Now, the use case typically involves measurement of in vitro data to hit the main mechanisms that are included in the software, then the prediction of exposure in the liver via PBPK modeling with GastroPlus, and next, sort of a layout of exactly what dosing protocols need to be run and the status of those patients in terms of being fasted and fed and the types of patients you want to simulate. Once those three components are brought together, we can predict the risk of drug-induced liver injury or DILI, but we can also determine exactly what mechanisms might be involved, the characteristics of the patients that might be responding, and we have risk mitigation strategies such as dosing changes or monitoring. Now you might be wondering, well, how can I access this excellent platform? Well, there are two different ways. The first way is by joining the DILIsym initiative. This is our consortium that has been ongoing for the last 10 years. The companies that join this consortium, which over the last 10 years has included 19 large pharmaceutical companies, receive access to the platform via 2 global floating licenses. They also receive the integrated GastroPlus version as one of those licenses if they choose when that version becomes available. They receive licenses to our high performance grid feature, which allows you to scale to as many nodes as you want for the purposes of your simulations. They receive a large discount on consulting, and most recently we've added all of these benefits for the DILIsym consortium as well for the price of one. So you get 2 consortium memberships for the price of 1. So we encourage you to join the initiative to obtain the software you're going to see today. Also note that you can acquire a single annual licenses if you prefer not to engage in multi-year contracts. Now, Corey will cover the interface today, but I wanted to mention some of the new exemplar compounds that we've added into version DSX. We've also added 2 new sim cohorts, which are very practical, smaller groups of simulated individuals that allow you to combine variability in liver injury parameters and biomarker parameters. For the exemplar compounds, we have a Pfizer compound 04895162, efavirenz, anastrozole, and tamoxifen. Now, for a couple of these, we have presented the results at national meetings, and I just wanted to highlight, this is a poster we did with Pfizer on the PF 04895162 compound, and you can note down here in the bottom left that we predicted this compound would be safe in rats, whereas it would have clinical DILI issues in humans, and that lines up well with what they saw in the clinic. And this example has actually been published since, so you can go and read that publication, or you can view this poster and the slides, and then you can play around with these parameters in the software once it's available. Another poster that we presented for one of these exemplar compounds is on efavirenz. And with this compound, basically what we see is that clinically, anywhere from 1 to 8% of patients have been observed to have ALT elevations, whereas in our simulations, we predicted 1 to 2%. So good alignment with the clinical data. Another great exemplar compound that you can use either for DILI DDI predictions or just to explore the software. Now, there are a couple of features of DSX that I wanted to point out before Corey takes it away. The first is the organization of projects. In the past, individuals created these sim singles or simulation setups, but you really created one sim single, another sim single. They kind of added up, in a menu, but there was no way to group those. So you may, for example, have a project for compound X, another for compound Y, and within each of those, you might have 20 or 30 different simulations, but they were all mixed together. With the new the new organization of the projects, as you see here, this DILIsym sample project list shown in the image, each project has a grouping of sim singles, a grouping of other types of options that have been set up, including Sympops and parallel run list and sweeps configurations. So you can group all of your, let's say, compound X work under one compound X project, and you could do the same for a different compound. So it's a much nicer way of organizing things. The other feature that's really nice is the parallel run list. In the past, this parallel list only allowed you to run sim singles in parallel, but now you can actually add Sympops or virtual population scenarios to the list, sim singles, as well as sweep configurations. So you could, for example, set up 2 different sweep configurations, 3 population scenarios, and 10 sim singles. You could set them all in one run list, and you could queue those simulations overnight. You come back the next day, they're all run and they're all saved. So it's really a nice way to mix a different types of simulations in one list and have them run in parallel. Okay, so enough of me. I'm going to now turn it over to Corey Berry, who's going to really walk you through a hands-on or a live hands-on demonstration of the software, which I think you'll find quite excellent.
Thank you, Brett. And before we do hand it to Corey, just a quick reminder that the video's audio feed will be played through the computer audio, your mic and speakers. If you are not hearing the audio properly, please use the hand raise icon for assistance. And let's get started with Corey. Take it away.
Hello and welcome to this Dillysimx video demonstration. Today we will be going over 8 features within the DILIsym software. These include SymSingles, Sympops, the run-in parallel feature, parameter sweeps, specified data, output panels, the command line, and also a demo of MitoSym. Now we will take a look at the DILIsymx user interface. When the interface loads, you'll be presented with a navigation bar on the left-hand side that provides you with different buttons to view different features within the application. Currently, we're selected the home view, and what this does is it allows you to see different resources online related to DILIsym modeling. So as you go through the navigation menu, you'll see that the contents of what's available changes. We'll be going over each one of these in upcoming videos. Now we're going to go over running a SymSingle and viewing the results. So first you want to go and select the SymSingle button from within the UI. If you have no configurations loaded, you'll be presented with this window. And so you want to click the Create New Configuration button and create DILIsym. So for this example, I'm going to be using the AMG 009 parameter set. So I'm just going to go ahead and give this name here for human. Okay, once that's loaded, you want to go ahead and select the correct drug parameter. And so within this window, let me go back for a second. When you select this button, you're presented with a pop-up window. Within this pop-up window, you have a list of different options, parameter sets, correct? So different from the previous versions, you can actually search within this list for what you're looking for. So in my case, I'm just going to put in the 009 on text, and you see that it filtered it for me. So I'm going to go ahead and select the human AMG 009 for the drug parameter set. So next I'm going to select the compound dosing. And again, I'm going to select the 009 human dosing for 100 mg. And for the time, I'm going to increase that to 2 weeks. All right. So now I'm going to go ahead and select a different output panel. I'm going to do all variables in this case. Note that this will produce more data, larger output files. And so now that everything is selected here, I'm going to go up to this Run button. You'll notice it's on the left-hand side of the actual configuration name. Click Run. So this is the actual run simulation window for DSX. And I'll go through explaining some of the parts of this. In the top left, we have the system status. This just gives an overview of how much RAM you have, how much disk space you have, and also how many cores you will be using from your CPU for this actual simulation. Since this is a SymSingle, we're only using one. We always use one for SymSingle. Here in the middle, we have the runtime control. This shows just the runtime of how long the simulation's been running. And we have this run button and this run and debug button. And the difference is the run and debug will actually perform checks low down in the solver that will slow down the simulation, but in case there's like an error, it will log that information and print it out here in the patient log when the simulation fails. So I say run that whenever you have an issue running the regular mode and you get a solver error, run it in debug mode and it should print more information related to that error. And here on the right side we have the simulation status window. This shows us how many simulations have been completed, how many have been saved to file, and how many have failed. And this is very useful for multi-run simulations such as Sympops and parameter sweeps. So moving on, here I have on the right hand side I have these boxes. This just previews to me what I selected from within the SimSingle. So for species parameter set I selected human, calories human. It just previews what I have selected, so I know, this is what I actually want to run. And of course, compound X dosing is set to AMG 009 as well as the drug parameter set here. So here in the middle we have a few buttons related to the SimSingle. We have, of course, a spec data button, a parameter input parameters button, initial states button. So these options will, once you click these, once they become available to click, they will give you information related to the actual inputs going into the solver. So since we haven't run anything yet, they're grayed out, but I'll show you what they look like when I click the run button. And of course we have the save results, view results button, as well as this copy and hide log. So the log is located here underneath the patient view, patient status panel, and it will provide you information messages directly from the solver as the simulation runs. So I'm going to go ahead and start this simulation. So just hit the start button here at the top. And as you can see, it's away. It's giving me a percentage. And during the initialization phase of the simulation, the parameters, initial parameters, and initial states were saved, so I can view those if I want to for this simulation. So I just show you what this window looks like. These are the initial parameters that went into the simulation and the initial states that went into the simulation. So now it's finished and it's saving. And so while that's saving, I'm going to show you what the stiffness report, well actually, we'll go ahead and save it first. So I'm just going to save here. And so what this stiffness button does is once you click it, it kind of shows you at different time points, how many iterations the solver had to perform. So we can see that at time point 253, it had to perform the most number of iterations. So we just say that was more of a stiff time point. So now that it's complete, I'm going to go ahead and click the view results button. And so this is the report viewer, a separate window that gets launched whenever you click the view results. And it's simply just a way to plot the data. So over here on the left hand side we have the list of all variables available, the time, vector, and all the output panel variables that I selected from within the SimSingle setup. And so to visualize the data, all I need to do is drag these variables to either independent or dependent boxes here below. So for this case, I'm going to drag the time to independent, so that it gets set for the x-axis. I'm going to select one of these variables to show for dependent. So let's see. All right, there we go. So, and you can just drag as many of these as you want down, and so forth. So, as you drag, it updates the plot. And over here on the far right hand side, you have the different options for changing the visualization of the plot, such as the font color for the axes, the font style, the chart color. So you can go in and select a different background color if you want it. And you can also turn on and off line symbols, the on plot legend, and turn off grid lines and things such as that. You can also set a a custom range for your axes manually here with these boxes. And also, if you go and hover over each axis individually and you use the mouse wheel, you can change the scale of that axis manually. So if you also wanted to view the data behind the scenes for this, you can click the table tab at the bottom, and here you have the, for the items you have listed in the dependence box, you have all the data associated with that for each time point. So you can save this data to a Excel file, or you can copy it to Excel as well. So this tool is actually still in development, so there'll be a lot more options added in the final release. And so if I go over here on the left hand menu and click eDish, you'll see that it plots the single point for this patient in the normal range. Alright, so I'm going to go ahead and close that. So I think that concludes just running a basic SimSingle within DILIsym. I'll go over more advanced SimSingle simulations in future videos. Now we will take a look at running a Sympop simulation. So in the previous SimSingle simulation video that I showed, we used the AMG 009 human configuration. We're going to keep this one for the Sympops. So one modification I will make to the SimSingle is to change the parameter set for the time to 24 hours instead of 2 weeks. So let's do that right now. Now I'm going to move on to the Sympops window, and as you can see here, we have no configurations available for Sympops. So we'll just click this Create New Configuration button, and we'll give it a name. Alright, so let's go over some of the panels in this view. So again, you have the configuration list here on the left hand side. This just shows you the different configurations you have defined for Sympops. Here in the middle, we have the Sympop Selection. This tells you how many patients are available in the currently selected Sympops that you have, which selected SimSingle you're using with this Sympops. This defaults to the whichever one you have selected in SimSingle. Also, we're looking to the, it lets you override which selected output panel you have for this Sympops. So I'm going to go ahead and select a different Sympops data to use for this. So I will use the multi-16A, which should be 16, for 16 patients. And for the output variables, I'm just going to select the common output to reduce the amount of data that needs to be saved. Alright, so you also have options to load the initial conditions for the Sympops, which is checked by default. I will leave that checked as well. And also I can select how many of my CPU cores I want to use for this Sympops. So I'm on a 32 thread machine, so I'm going to go ahead and just use 16 since I have 16 patients here. So if I hit normal, that'll use half of what I have available, which is 16. Alright, now that everything looks good, I'm going to go ahead and click the run button for this Sympops. Alright, you will see that this is somewhat similar to the run window for SimSingle, but we have a few different changes. So first of all, the system status now shows us using 16 of 32 threads. And over on the simulation status, which shows us 0 of 16 completed. So that kind of reflects that we will be running 16 simulations for the Sympops. Again, we have many of the same preview boxes from this SimSingle on the left hand side and right hand side. Instead of this spec data, we have which Sympops we have selected here at the bottom right. Now in the middle, we have perhaps the most changes to this view, we have this new visualization for showing different simulations running on different threads. And you will get a better idea about this when I kick off the simulation. And here at the bottom, we have some options for viewing all the results for the Sympops and also for showing the patient. So if I click show patients now, you'll see that it just shows which variables, the patients and the variables that are stored within the Sympops. So I'm going to go ahead and kick this off. So hit the run button. And so this is utilizing 16 threads in parallel. And as you can see, as it runs, it's red. When it goes this dark color, that means it's completed. So far I've completed 9, or actually 11 of these simulations so far. So this is a pretty fast machine, so it kind of goes through those pretty quick. So again, once it completes, I'm presented with a save results button. I'm going to go ahead and save this. All right, so now I can actually click on these individual runs, and I can view the logs for each one individually. And I'm presented with the same options that I have for SimSingle, with viewing the initial parameters, initial states, and so forth. So I can scroll up and see what the log looked like for this individual patient 15 simulation. So I'm going to go ahead and click. So there's a couple things I can do. I have this sim report view. If I click that, that goes through and shows, which simulation was the fastest, which patient had the fastest simulation, the slowest, which has the most number of iterations and the least number of iterations. And if I scroll through this table, I have this status column which tells me whether or not a patient completed. And also I can view the stiffness report individually for each one of these. So just click one of those. You'll see for patient 3, this is the stiffness report. At time point 11, it had perhaps pretty high number of iterations. So let's close that out. So I'm going to go ahead and view all the results for this Sympops. All right. So for Sympops, we have this a special view, slightly different from the SimSingle report viewer view. So when you click these available variables, and again, I only have 85 because I selected the common outputs. When you select one of these, it's going to plot all the patients for that particular variable. So I'll go ahead and select, let's see, CLSFA. Yeah, so in this case, it's rendering all 16 patients. And I can kind of browse this as I wish and kind of scale it up or down for each axis. Again, we're given the same options as the SimSingle view for modifying the way the plot looks. And again, we have a table view in which we can see, for each patient for this particular variable, we can save that data as an Excel document or copy it out as well. All right. So if I click the legend button here, I'm given a view of each patient and which color it is. So, and I can also go in and select one of these lines from the graph, and it tells me which patient it is. So if I go to the eDish, I can see that a lot of these kind of line up and appear as one dot, but it's actually multiple different selections for the eDish. But they're all within the normal range. All right. So I do believe that concludes running a Sympops. As you can see, it's pretty similar to running SimSingle, except you have a good bit more data to look at and slightly different way to visualize it. So I think the next one we'll talk about would be the running parallel list. Now we will take a look at running the run in parallel feature for DILIsym. So in the previous parts, we went over SymSingle and SymPops using the AMG 009 parameter set. So under the parallel button, you can see I've already created this. So I'll go ahead and delete this. So I'll go ahead and create a new configuration. And so what I have here in this available modules box, it gives us a list of all the available runnable configurations within DILIsym. So since I've only created 2, there's only 2 here. So I'm going to go ahead and add all of these to the selected modules box here, so that whatever's here will appear in this actual running parallel configuration. So now that that's selected, I'll go ahead and select here at the bottom how many threads I want to use for this running parallel configuration. So if I click the low key here, it's going to select 8 of 32 for my machine. The low only use 1/4 or 25% of what you have available on your machine. So now that that's selected, I'm going to go ahead and click the run button here for kicking off this parallel configuration. So what you'll notice that's different about this run simulation window is we no longer have the preview boxes on the left and right hand side because we're selecting from multiple, could be totally different configurations. We don't necessarily need to know what the preview boxes were for each one of those. It's just a collection of them. And you'll also notice in the simulation status, it shows that we have 0 of 17. That 17 is a combination of the multi-16 Sympops and the single SymSingle simulation. So it always keeps track of the total number of runs that you have, regardless of how many Sympops or parameter sweeps you may have selected. So I'm going to go ahead and kick off this with by clicking the play button here at the top. So also note that the order that these run in are based on the order that you have them stored in the selected modules list when you create the actual configuration. All right, now that those have completed, I'm going to go ahead and create a new folder. So what happens with the output with the parallel run is that when the output results complete, when the simulation completes rather, it will ask you to create a folder to save the information to. So I created this out folder. I'm going to open it up and it's going to save it to that folder. So now that it's saved, I can go ahead and click the view all results button. All right, so the view all, for running parallel, you get another pop-up box when you go and view the results, because this is a collection of different runs or different simulations. You have to select which one you want to actually view from within the output results. So in this case, I'm just going to select the Sympops one. And there we are. We have the Sympops for this run. So I can go ahead and just select any one of these options and go ahead and view it. And so if I wanted to go back and view all results again, I could select the AMG 009 output results. And this gives me the original SymSingle plot view, where I can drag the time variable down, and now I can just select these individual output variables. All right. So that's pretty much how running parallel works. You're just combining a list of different configurations together to run them in parallel. And it breaks them for those multi-run simulations, such as Sympops and parameter sweeps. It'll break them down into individual runs to be ran across multiple threads. And so I think in the next tutorial video, we'll go over running parameter sweeps. Thank you very much. Now we will take a look at the parameter sweeps feature in DILIsym. First, click on the button for parameter sweeps and create a new configuration. Okay, so here's the new user interface for parameter sweeps. I'll try to describe some of the new panels located on the right hand side. The select base sim single is pretty obvious. You click it and you can select from a current list of sim singles that you have defined. The select variable allows you to select the individual variable that you want to sweep across. So we have a new user interface for finding a variable as well. You can still locate by group and subgroup. So if I go through and just click these different parameter groups, you'll notice that the subgroup category changes, and if I click the subgroup, it changes which variables are available. If I leave all groups and all subgroups selected, I can also go over to the third column and search by a variable, and because this list will contain all the available parameter values. So what I want to do in this example is just I want to sweep across the total simulation time, just as an example. So I'm going to select total simulation time and select variable. So here in the sweep options, I have the lower value, the upper value, the number of steps, and also the base value. So in this case, I'm going to just change this slightly. I'm going to start at 300 for the lower bound, and I'm going to go to 330 for the upper bound, and I'm going to change the number of steps to 30. So now I'm going to click this plus symbol, which will generate a column to the sweep table. You will notice that the table now contains about 30 values sweeping across the total simulation time. Now if you see the option step through all variables simultaneously, when this is unchecked, every cell within the table will be considered a simulation. When this is checked, every row will be considered a simulation. For now, I will leave it unchecked. Moving on to the core count selector, in this case, I'm going to select normal to use half of my available CPU threads. Now I'm ready to run the parameter sweep. I will go back to the configuration to the left of the configuration name and click the run button. Alright, in this case, this looks very similar to the Sympops. I have these selected sim single preview boxes on the left and the right hand side. And I also have the total number of simulations and the simulation status here at the top of 0 of 30. So I'm going to go ahead and click the run button. So now we have each one of these simulations, starting with the first one. It should simulate between 300 and 330 as the total simulation time. I will now speed up the video for the sake of saving some time. Alright. So I'm going to click the View Results button. All right, I'm going to go to the table. And if I scroll down, you'll notice that the time column should have all the different times selected from 300 to 330, and it does. The last one here is 330. So I won't go through anything else. That's just a quick explanation of how the sweeps feature worked. Now we will take a look at using the specified data feature in DILIsym. On your navigation panel, scroll down to the specified data button at the bottom. You'll notice that this UI has altered quite a bit from the previous DILIsym version. And I'll go through explaining some of the new panels located in this user interface. First, you have a group and subgroup panel at the top. This allows you to find state variables using their group and subgroup names. By default, all the variables are listed here in this panel underneath the group and subgroup panels. And you can also just filter by name if you know what you're looking for. All right. So when you're ready to create a specified data template, you basically select which variables you want to have in the template, and you can hold down the control key to select multiple variables. And next you'll hit the arrow key that's pointing over to the right hand side. Once you click that, you'll have the option to add these variables as constants or as time dependent variables. In this example, I'll do constants. And now I'll go back and select a couple more variables, and we'll do these as time dependent. All right. Notice at the top you can select the number of individuals you want to include with this specified data template. I would change that to 5. At this point, I'm ready to create the template. So all you need to do is click the Create Template button located at the bottom right. We'll go ahead and give it a name. So now you'll be presented with this pop-up that tells you you've successfully created the template, and it asks you if you would like to open it in your default spreadsheet program. For most people, this will usually be Excel. So if you do want to open it in Excel to see the template, you can click yes, or you can just click no, but you can still open it later, and I'll show you how to do that. For now, I'll go ahead and click no. Alright, now we've added this template within DILIsym. There is another tool called the Data Manager. If you click this button, it'll show you all the different specified data templates and files that are included with DILIsym. And if you scroll all the way down this, you'll see that you've added your template file to this list. And from here, you can go ahead and open it using this second icon at the far right. So I'll go ahead and open it from here. So this opened up in my default spreadsheet program, and you'll see it's just an empty template ready to have data inserted for your specified data. Okay. So that really concludes creating a specified data template, and you can create as many as you want, and you can even open previous specified data templates as well and modify those. Alright, now I'm going to go back to SimSingle, and you'll see there's a specified data box at the bottom right. This allows you to select from one of the specified data templates embedded within the application. So what I'm going to do is select the one I just created. And if you scroll down, you'll see it's my template here. Now that that's selected, I can go and run this simulation. So of course, in this case, it's not going to really do anything because I haven't added any data to it. But I just wanted to show that once you run the simulation, you'll see that the specified data preview field has my template in it here in the bottom right. And if I hit run, yeah, of course it'll error out in this case because I haven't added any data. But if you have added data, it'll populate and you'll be able to view which data was added with the specified data button. So I'll actually do that. I'm going to go back and select one of the data fields that's been populated. So now we'll try to run again. All right. So that's going to save. We can save it, and now you'll see here that we have the specified data button feature, and we click that, and this just provides a detailed log of when at which time steps each specified data variable was changed, and it shows it before the solver steps and after the solver steps. So it's kind of a way to track and make sure that that information is actually getting reflected in the solver. All right, I think that concludes the specified data feature. Thank you very much. Now let's take a look at creating an output panel. So I'm going to go ahead and log into the software. On the navigation panel, just go down to the tab or button that says Output. And from here, you have quite a few panels to manage and select and create output panels for your results. So I'll describe some of these panels in more detail. The Select Group panel here at the top allows you to select different groups, algebraic groups or states. And this Select Subgroup allows you to select the subgroups from that. You can use these options to kind of filter through what variables are available, or you can just go directly to this text box. If you know kind of what you're looking for, you can just type it in and it will locate it. So in order to create a new panel, just go down at the bottom right hand side of the window. There's a Create Panel button. Just click that and give it a name. So once you create the panel, it's initially blank. So what you want to do is start adding different variables to this panel. So in my case, I'm going to add all these GSH variables to the panel. So I'm going to hold down Shift to multi-select, and then I'm going to click the arrow pointing over to the empty box. All right. So now that that's been added, I'm going to go back to my Sim Singles, and I'm going to select a different panel from the list. And see here at the bottom, you see my panel has already been added. So I'm going to go ahead and select that. So everything else looks good here, so I'm going to just run this simulation. All right, now the simulation is ran. I'm going to open up the results. And as you can see, it only includes the variables that I've added to that output panel. So I'm going to go ahead and see if any of these have actual data related to them. Yeah. So that's essentially how you will create your own custom output panels in the new version of DSX. It's pretty simple. And let me go back to the original list. And so we also have this panel where you can select from existing built-in output panels, and you can see that these have already been built and ported over from the previous version of DILIsym. All right, so I think that kind of concludes the output panels feature for DSX. Thank you very much. Now let's discuss running DILIsym in command line mode. So the command line version of DILIsym is capable of running two kinds of files. .run files, which are produced from the DSX GUI for SymSingles, Sympops, parameter sweeps, and optimization configurations. And also the run list file, which is produced primarily from the parallel run list feature. So the way this works is in the command line mode, well, the first step that you need to do, you export one of these file formats from the DSX GUI. And then you will launch the DILIsym command line, sending in the correct parameters that point to this run file or run list file. The simulation will read that file, determine what kind of simulation it needs to run. It will then execute the simulation or simulations, and then it would save the results of the simulation back to the same run file. This is the default behavior of the current version of DSX. So here is a quick overview of some of the common arguments for the DILIsym command line. There are many more than this that will be documented in the final version, but here are some that are perhaps the more important ones. First, you can override the number of threads to use for the DILIsym command line to use for simulations. By default, this value is read in from the actual GUI. When you select the slider in the GUI to select the number of threads, it's read in from that value, but you can always override that directly. Next, we have the run config file, the actual location of the run or run list file that you'll need to send in. You have a couple flags here. You have a verbose flag that you can send in as an argument, and this will print more output for you. And also there's a debug flag. Now when you're in the, I've talked about this some before, but when you're in the actual GUI, there actually is a debug button, and when you click that, extra checks are ran behind the scene for the solver to kind of troubleshoot any kind of issues you might have. Behind the scenes, this is actually just calling the command line version in debug mode. So you can also manually send this in to the command line version as well. So now what I'll do is I'll go through an example of exporting a run file, Sympops in this case, and executing it at the command line. So I already have a multi-16 run ready to go. So I'm going to select the one that's highlighted here. So instead of clicking the run button to execute it from the UI, I'm going to hit these three dots. This option menu will pop up, and you'll see there's an export command here. So I can export this Sympops to a single file. That is the option that you want to select. So I'm going to just export this to the current window. All right, now that it's exported, I can close the GUI out completely. Now, I'm going to go open up a command line shell, and I've already entered the command that I actually want to run. So I entered the DILIsym command line executable, and then we enter the dash dash threads, as I mentioned before, as one of the arguments. I'm going to set this one to, actually we just set it to 8. And also I give it the actual run file for this run configuration. And I'm going to tell it to print out more output. So now that everything is entered, I'm going to press the return key and kick off the simulation. And so while this is running, I'll go over some of the output that gets generated. So when you kick off a sim with verbose mode on, it prints out quite a bit of information pertaining to the run that you're executing. So you get the name of the run, and this also is shown in the actual UI. You get the UUID, the type of run. This is a Sympops. We get the app name, the version that's running, the time we kick the run off, the date, the user, the person who actually kicked the run off, and also the name of the file again that we're kicking off. And so what we see here is that, the first thing we do, since this is a Sympops, it's basically extracting that Sympops to individual sim singles and then kicking off each sim single as a simulation across 8 different threads. So as you see here, this is all the things executing in parallel. And so here at the very end, we see that the entire process took approximately 41 seconds to execute 16 simulations, running with 8 threads. So there will be more additions and different arguments you can pass in to the command line mode, but for now, this is a basic example of how it works. Alright, and so in that case, I'll just shut down this video. Thank you very much. Now we'll go over running MitoSim within the DSX GUI. So I'm going to go ahead and log into the software. So the first thing you want to do is go to the sim single option on your navigation menu. And down at the bottom left, the create button, click it, and you'll get an option to create both either a DILIsym sim single or a MitoSim sim single. Go ahead and select the MitoSim. So as you can see, the MitoSim gives different options that you can select, different parameter sets that differ from what DILIsym offers. And this is how MitoSim is integrated within the app. So I'm going to go ahead and select a few options here to simulate. Alright, now that I've selected those options, again, just like with DILIsym sim singles, I will click this icon to run the MitoSim sim single. As you can see, we also have preview boxes on the left hand and right hand side of this window to let us know what all we selected for this MitoSim sim single. Everything else still remains the same as with the DILIsym one. So now I'm ready to actually kick the simulation off. So just press the play button above. And once the simulation completes, we'll hit save. You notice we have, the same kind of log generated from the MitoSim. And you can click the view results to open up the output. So now the output is loaded. I'm going to go ahead and see if I can render some of these charts. All right, so it appears some of this chart data looks okay. So yeah, this works exactly the same as DILIsym, except it's just running MitoSim in the background. So I think that will conclude this demonstration of running MitoSim. Thank you very much.
Thank you, Corey. You provided us a very clear picture of what we can expect. Before we begin the Q&A session, we have a quick question. Which sector best describes you or your work? And if you haven't already done so, you may send your written questions using the questions pane on your control panel. Or if you'd like, you can send it via chat, whichever option is easier for you. We'll give this poll just a few more seconds. And while we do that, I'd like to now introduce Scott Seiler, Chief Scientific Officer at DILIsym Services, to moderate today's Q&A. Scott,
Hi.
take it away.
Hi, everyone. Thanks for, and thank you, Arlene, for the introduction. Thanks for joining our webinar. We do have a couple of questions, and we're ready to receive as many as you're ready to deliver. We'll start off, and actually, I think Brett Howell will be answering each of these questions. So, Brett, here's the first question. Does Dillysimx provide an enhancement to simulation performance?
Yes, actually, Dillysimx will be considerably faster than DILIsym version 8a.
All right, so question number 2. This is related to the command line version that Corey just described here at the tail end. Since there is a command line version, is it possible for someone to obtain permissions to deploy the simulation engine to the cloud for a large number of nodes to be used?
Yes, absolutely. I'm glad you asked. That we have what's called a high performance grid license option. And this is an additional fee that adds on to at least 2 end user license purchases, or it's included if you join our consortia. And it allows you to scale the simulation engine, the command line format, in other words, of the software, to as many nodes as you like. So there's no limit on that scaling, and that can be done to a cloud-based system or your local cluster. So it's a great feature.
Looks like there's one more question, unless there's one that comes in the last minute. This is related to performance as well, which is, and the question is, will the Sympops within DILIsym run more rapidly in DILIsymx?
Yes, all the simulations, Sympops included, will be faster and more efficient. Our initial speed tests show dramatic improvement. And in particular, the Sympops will be much faster because the parallel functionality, the parallel tasking out to different cores and different threads is much more efficient in C++. So it's a great improvement.
Well, that was the last of our questions. So with that, I think we can probably wrap up. Brett, why don't you say your farewells, and then let's wrap up this webinar.
Okay, well, thank you everyone for joining us today, and we really hope you'll contact us about getting access to DSX and using this great software to improve the safety and efficiency of your development programs. Arlene?
Thank you, Scott, Corey, and Brett. You have definitely shown us how DSX utilizes various data types to make better informed decisions. To test drive DSX and all the latest features for yourself, check us out at www.simulations-plus.com. This concludes our webinar for today. It has been recorded for playback and will be available on our website and YouTube channel. Thank you very much.
DILIsym Services, a Simulations Plus company, provides powerful science and innovative technologies to support research and regulatory interactions. DILIsym version 10, also known as DSX, is quantitative systems toxicology software capable of predicting and explaining drug-induced liver injury. This version offers a brand new user-friendly look and feel. DSX is designed to provide an indication of the potential DILI hazard posed by individual molecules and to provide deeper insight into mechanisms responsible for observed DILI responses at various stages of the drug development process. Predict hepatotoxicity for phase 2, 3, or 4 clinical trials in patients, first-in-human clinical trials in healthy volunteers, small and large preclinical species, and inter-individual variability in populations. What's new in DSX? Contact us today to save time and resources on DILI hazard research. Special programs are available for instructors, students, and researchers. Learn more at simulations-plus.com/DILIsym.